All right, why don't I kick it off? Hi, everyone who's here right now in the present and all those people joining us in the future on YouTube. It is my distinct pleasure to have our distinguished guests here to talk about fabrication in the body and all the things digital fabrication can do related to health. Our first speaker is going to be Albert Folch, correct me if I'm wrong. And the goal of his lab is to make microfluidic devices that are as easy and intuitive to use as smartphones. He's developing the next generation of microfluidic devices for applications in automated cell culture, neuroscience research, cancer diagnostics, and cancer therapy. Before coming to UW, he was a postdoctoral associate at Harvard and a postdoctoral fellow at MIT. And he is an American Institute for Medical and Biological Engineering College Fellow and a recipient of a NASA Space Act Award and an NSF a Career Award. So with that, Albert, it's a pleasure to have you, and we'd love to hear what you're working on and what you want to tell us today.
Thank you, Jeffrey. So should I just go ahead and share my screen then?
Yes, please.
All right, let me try. I think it should be fine like this. Do you see it now?
Yes.
Perfect. All right. So I'll
just want to switch to slide presentation mode. There we go.
Yeah. My lab works on general interface of microfluidics and cancer, as you mentioned. But today I'll talk about our efforts in 3D printing, because that is what affords us the ability to prototype our devices in the first step of our translational efforts. And I think I like to put this slide as a motivator, because one thing that strikes me as in the world of digital manufacturing is the fact that we are surrounded by a lot of objects that are very important in our daily lives. And there are now, a lot of industries use digital manufacturing to go from idea all the way to fabrication. And they use computers to plan and execute the fabrication all the way using the computers. And but in microfluidics, we don't use that paradigm of fabrication. And so that's what my lab started identifying, this shortcoming early on. And we tried to see if we could use digital fabrication specifically for microfluidics. And one thing we focused on is in using, getting lessons from tissue engineering to see if we could take, there are already very nice materials, most notably PEGDA, polyethylene glycol diacrylate, which if you mix with, because it has two acrylate groups in the end, and based on the molecular weight of the monomers, when you add the photoinitiator, you can get either a hydrogel if you have a large molecular weight that associated with water during the polymerization process, or if you have a low molecular weight compound like PEGDA 258, then you can have essentially a plastic. So that affords kind of like a range of materials that is very appealing for as a starting resin for especially for stereolithography, which is in itself limiting in terms of materials that we can, the range of materials. But this affords a variety of materials, because this is non-porous, so very useful for making the bulk of the microfluidics, and this is porous. And as you can, as I'll show you, we make microfluidic devices that contain both types of materials. We started by early on by verifying that we could build PEGDA 258 devices, these plastic devices, to show that they were cytocompatible even with very delicate cell types like neurons, and also more biotechnology cell types like CHO cells. And we saw no difference on these surfaces after the thorough washing treatments between that and tissue culture polystyrenes. And also we were able to using very smooth surfaces like glass, printing against glass surfaces, we were able to get transparent devices. So that's an important thing that we did very early on to verify biocompatibility and for printability of our devices. Another thing that we worked on to improve the processes that were out there was to add, we typically work with your A19 because it's very cheap. We get like a bag like this we got it as a donation. The companies that manufacture this, I believe it's BASF, would not sell it to us because they sell it by the ton, and they for painting. And so we said, oh, can we have a kilogram? And a kilogram will last us a couple of centuries. We calculated because we use so little. And then we worked on adding ITX, which is essentially it's a photo absorber. It is also that helps in adding in absorbing more, essentially in increasing the absorption at the lamp's wavelength. And so that also added an element of resolution in Z especially. That was very important for creating the roofs in microfluidics, and that helped us to create high resolution prints in stereolithography. And you can see here a variety of structures that were that mixtures allowed us to create, such as single pixels. You can see this is the mirrors from the DLD. And then we had enough the resolution, and the reaction was so fast that we could even see an image from the pixels. You could see the dimples from the pixels and then the marks from the. So that's what's given by that reaction. And you can see also channels that were 40:1 in height and then single pixel in width. And so because our projected pixel wide width is 27 microns. By the way, the ones that you see here are not the fault of the printing process. They're actually pixels that are broken in the STL file. There are problems in the STL files that need to be addressed by the industry, but that's something that we cannot address. We were also able to print things that are not moldable, well, this one is moldable. We printed it just to show more complex printers, but there are things like a double helix mixer or a coiled device like this. These are things that cannot be made in devices like this, cannot be made in PDMS and so on. Then we went on and also showed that we could build valves with this is all integrated, by the way. So it's all printed in one shot. So there's no layering or anything. And I believe these are movies I can play for you. So you can see that these are valves that we can actuate, and they're pneumatic valves because they contain a little membrane here that you can open and close. So it's very reliable. We can also we can add additives to the membranes to make it more make the PEG D8 hyperelastic. Or we can also, like I was mentioning before, because these are acrylates, they are compatible with each other. So you can start building with a PEG D8-58, which is a plastic, and then switch to a resin that is a higher molecular weight PEG DA, like the 700 molecular weight, and that creates a porous structure within the channel, and they bind to each other. Then you can finish the structure with the plastic, with a PEG D8-258, and when you finish your channel, it's all embedded inside, and so you've created a barrier inside between the two channels that is porous in between. And we verify that as you fill one of the channels with fluorescein, if you fill this one, for example, with fluorescein, you would end up having fluorescein on the other and on the other channel by diffusion. So that's something that you can you could of course build this device by PDMS by several layers and so on. But the elegance of this approach is that it only takes an untrained operator to switch the resins, and the whole the whole alignment of these layers is done by the program by the it's all done out by the machine. And we were also able to print PDMS because you can buy monomers of PDMS that are acrylated, and we mixed two types of monomers, different with the acrylate groups on different ends of the molecule, and then we found a good photoinitiator that was soluble in PDMS, and we were able to print PDMS in a way that was very transparent and with very good mechanical properties, very similar to Silgard 184, and build PDMS channels as well with decent resolution. The resolution is not as high as with the PEG D8-258, and that's because the resin itself the reaction is slower, so you will never get as high a resolution. So this is all I have for today. I believe I have probably almost over my time, but I'm happy to entertain questions, and these are sources of funding. Always thank you. So I'll, if there are any questions, I'm happy to answer them.
Excellent. Let's give a round of applause.
Should I stop sharing now?
Well, let's see if there's some questions from the audience that your slides might be useful for.
Okay.
There's no questions on the chat yet, but I was wondering if you could talk to the like maturity of this technique. Yeah, kind of where it stands right now in the field.
Actually, we are working with a very advanced 3D printing service. It's called BMF, they're based in Boston, and they're very interested in these resins and in the PEG DA one, and also the one that is very elastic. So we're working with them to make it more available, and we're working with them to facilitate the translation of these resins to them, because we believe that they should be they're actually open access. So the formulations you can't patent PEG DA. But even so, that they were very interested in it, just because they've been having trouble making things biocompatible, and they were interested in the fact that we seem to be able to culture cells on these materials. So they were able in that aspect of they wanted to reach out to the microfluidics community. So yeah, in terms of the technology, the resin technology, I think it should be easy to commercialize.
And I don't know if someone else had a question, but I was also wondering with the machine compatibility, like, do you need a specific type of SLA printer? I don't think you mentioned that.
No, these are, we work only with commercially available, like these desktop machines. We work with an Asiga printer, but we've also printed with, we actually started this work with another printer. So there's no requirement. We usually use machines that work with 385 nanometers. So it's a light requirement. But that's given by the photo initiator. And that's because we use IrgaCure 819, and that's the resin. So that resin has to go with that light, and usually, but so that's no, there's no specific requirement for a machine per se. Yeah, these are open source resins.
Great, thank you.
I have a question, if you don't mind.
No, yeah, go ahead.
So I was very impressed by the slide where you showed the individual features of the DLP mirrors showing up in the resolution of your prints.
Yeah,
that's kind of amazing resolution. Do you think you talked about some of the improvements in chemistry? Do you think there are some more improvements with respect to optics or hardware? Could you get even better resolution? Do you need better resolution?
Oh, I think I'm pretty sure these are, yeah, I'm pretty sure we haven't hit the highest resolution. These are, we happen to have 27 micron resolution, but other people see pixelation also when they print with these resins. It's not unusual with acrylate resins to see this type of pixelation. I would say, I usually say to people that if you do when you're trying to print a plastic, okay, with a hard plastic, if you do not see the pixelation, that means that that is usually something wrong with your chemistry and machine combination, because you should be able to always see the resin should congeal, cure fast enough to see the pixels. The machine is designed to create that at least a few layers. You should be able to see that pixelation, and if not, that means that there's something wrong with either the focus or that the resin is if the resin is diffusing too much for another reason. So that's always a good test to try to see if you see these pixels.
I see. Thank you.
So our next speaker is Dmitry Levin. He is the Associate Director of the Cardiovascular Innovation Center. The center's main focus is on the development of cardiovascular devices for diagnosis and treatment of coronary and peripheral artery disease, structural heart disease, heart failure, heart rhythm disorders, and data obtained from echocardiograms. As you can tell, I'm not a medical person. And other imaging systems, and translates the data into three-dimensional 3D printed organs. This allows physicians, fellows, residents, medical students to learn from accurate 3D models of patient-specific anatomy for educational settings, as well as supplementing patient care decisions for procedurists. He is also a former director of the UW COVID-19 Innovation Task Force. And from that experience, I got to see him as one of the hardest working people I've ever met. And so it's a pleasure to have Dmitry here today.
Well, thank you, Jeff. Appreciate it. Thank you for the invitation, first of all. And it's definitely been interesting to work with the entire UW community from the COVID perspective that was just mentioned a couple seconds ago. So I'm going to try to keep this light. I think the presentations were very much so structured nicely in that pattern of things. And what I'm going to do is I'm going to step out a little bit from the micro scales that were described in the previous two conversations, and step up a little bit more to the medical aspect of things. And then maybe the last presentation can come back to the aspect of things that we're interested from the bioengineering. So why 3D printing in medicine? And we've had some conversations about that. We've had the idea about bioprinting organs, about the idea of microfluidics and treatment of patients. So one of the goals that we've tried to accomplish is really frankly simple, but complicated at the same time. How education and case planning and patient treatments have been done 1,000 years ago to 100 years ago to, I don't know, 3 years ago is pretty much the same. This hasn't changed. It's the idea of getting together with your fellows and going over the cases, learning the technology, and it's idea of ongoing learning of anatomy. So when it comes down to treatment, what you're doing with the 3D printing is you're reducing some of that risk and eliminating some of the stress by putting that type of teaching, training, and education and device development in the low stress environment. Unlike patients where you're dealing with an N of 1 every single time, within the cadaveric studies, you might be able to replicate it multiple times potentially, but it's finite. With the 3D modeling and part of the digital modeling as well, is you can really think on the much grander scale and replicate the results over and over and over again to make sure you're getting to the proper result at the end of the day. Now, what are some of the things to think about is just like the engineering, we have X, Y, or engineers have X, Y, and Z scales. In medicine, we want to make things complicated. So we're going to call it coronal plane, sagittal plane, and axial plane. And now to really make sure that we can keep our job, we're going to add a couple more letters in there and numbers just to make sure no one knows what anyone is talking about. And this is all done on the digital scale. So you're looking at the 2D slices, one slice at a time. And for those of you who have seen this, I'm sure you know what this is. For those of you who have not, here's a little test. What do you think this is? So this is an example of one of those planes, the axial plane. I'm gonna step forward to a sagittal plane. That's the next one. So I don't know if now this is starting to become a little bit better for people and thinking what it is. And here's a coronal. So we kind of have an idea of what it is. Maybe you understand what it is. Here's what it is. And people have to put this together in their brain, and they have to translate that two-dimensional slices affiliated with it into a three-dimensional object that they will actually be working with, in this case, a patient's anatomy. So computationally, giving someone a 3D model is much less expensive than asking them to translate that two-dimensional slices into something else. So what are the pains? The pains are threefold. The pains are, what is the data that we're working with on the medical field? What are the materials that are available to us? And what is the equipment that's available to us? For those not familiar with the 3D printing, there's about 7 different technologies that are involved within the different additive manufacturing. Medicine can easily use 5 of those on a daily basis based on the applications that we're trying to address. So the pains with the data is, unlike the traditional manufacturing where you start with a CAD file, then you maybe translate that into an STL file, and then you translate that STL into a physical model, we're making things more complicated. We're starting with a DICOM data set, and the DICOM data set is a volumetric data set. It's not a meshed, it's not a surface-based data set. That means we have to go through what is called segmentation and sort of separate the parts that we want from the parts that we don't want by basically outlining, let's say, a kidney or a rib or a heart and saying, these are the parts that we want, and we don't want to print anything else. Otherwise, the printer is not going to understand this. So this creates our initial STL file. Then we have to go into CAD and do the next set of steps and frankly, clean it up a bit before we can go to STL and get the physical model out of it. And the DICOM data sets come in multitude of varieties. Here are some of the 4 common examples that you might see. On the left-hand side is a CT. And then we have the MRI. Then we have what is called a transesophageal echocardiogram, or TEE, and a set of electrophysiology EP studies. Each one of them can be translated into a 3-dimensional model, but each one of them comes with a set of challenges. Between all of them, the most common thing is the fact that we work with what's called the Hounsfield units, similar to the grayscale units that are available. But each data set has different resolution and different ability to manipulate those Hounsfield units or the grayscale in order to get us where we need to go. So here's an example of traditionally how medical 3D printing works. Here's a CT data set that you can manipulate, usually working within all 3 dimensions. So the X, Y, and the Z scale, or coronal, sagittal, and axial. And what we're doing is a common technique called the thresholding. Thresholding allows us to separate a set of grayscale values or Hounsfield units that we're interested in from everything else that we're not interested in. Once that's done, we can start subdividing those areas into multiple parts. So this way we have more areas of interest that we can then potentially transition and print on multiple materials based on what the interest of each one is. And on the bottom right-hand corner, you're gonna see that example of, in this scenario, parts of the heart that were separated into 3 different parts. In this case, we're working off of the CT, and we're working off of the blood pool. So really you're looking at the internal parts of the heart, not necessarily the vessel wall or the atriums and the ventricles and the external aspect of things. So that will have to be adjusted for. And that adjustment happens just like you would do in the engineering world via the CAD. So it can be smoothing, we can be correcting some of the holes and openings in order to make it printable. We could be mirroring objects, creating a wall because we're working initially with a blood pool and really we're interested in the internal aspect of things versus external in this position. And then creating a sliced object to allow for better visualization. And this is great, but it creates another set of challenges. If this is our initial blood pool and we start working with it and exporting it out, we can either make it hollowed out and extrude inside, or we can extrude outside. And depending on what we're doing, we're going to be changing the overall geometry of that original object. In the end, maybe ended up with something like this, which is pretty different from the original blood pool that was started with. So we have to be careful in the steps that we take and making sure that we're not manipulating the original data set, that original anatomy that we've gotten through the imaging modalities. Now, similar to what is being done in majority of the 3D printing, there's the step of the post-processing. And important thing to remember for anyone who's interested in 3D printing, and especially people who are interested in medical 3D printing, that step is very time consuming at times. So just because you've made a part, which took you time, printed a part that took you time, you might be spending just enough time as the first 2 steps to post-process it. And this is an interesting graphic because it shows some of the challenges that people can think about when it comes to post-processing, and different technologies have different problems. Some are faster, some are slower, but overall, it is definitely a consideration for people who are trying to make things. With that, we're stepping forward into maybe a different permutation of things. Is similar to how anything functions in the medical field. We need to make sure that we're verifying and validating our models, and we're actually creating a good model that is not just accurate, but is also applicable to the particular case that is being done. So we've identified about 7 different steps within the 3D printing process for medicine, and each one of them comes with a separate set of challenges. For the example, for step number 2 of the segmentation of the anatomy, we might have an inaccurate segmentation, or let's say in the post-processing part, we might be under-curing or still having a green part. We might have some material deformations through heating and autoclaving, or it might simply be a damaged part at the end of the day. And then with that, all we've really done is, we've created a part. Now, that part is not final yet, and it's not final yet because now we got the next set of challenges, which is device status and classification. In medicine, there's sort of these 4 different classes that people have outlined, starting from class 1, which is the lowest, and going up to class 4, which is the highest. And each one comes with a set of challenges and a set of regulations. So even though this graphic is very busy and kind of outlines a lot of the processes that are affiliated with 3D printing and in medicine, it's really that bottom line that I want to highlight for everyone, that even though you're making a part, you still need to make sure that part, because it comes down to the patients, is clarified, it's validated, it's qualified, and it's actually usable from not just perspective of being the visual part, but if you're making an implant or if you're making something that's going to come to patient contact, that is also important things to consider. So those were all the negatives. Now, at the same time, there's lots of positives that come with these things. Why do we do this? Because we do it for patients, we do it for physicians, and we do it for outcomes. We do it because the 3D printed models can improve all of those factors and really reduce a lot of stress on physicians. They can increase the understanding for patients to know what is going to happen to them. And we can go and make cases that we weren't necessarily able to do before. So here's just simple examples of some of the models that were done. As more flexible materials and more elastomeric materials are coming to the market, as more people are looking at multi-material printing, as more people, similar to what Cole has been doing, are looking at a variety of ways to be able to easier process that data set to get us closer to what it is, then we're able to frankly treat more patients. And here's an example of another case, a very complicated surgical case that you need to redo on the left-hand side, and each part is in different color for each different anatomy in question. And on the right-hand side, you can see a sample implant that the physician was planning for and trying to understand both the approach, the size, the complications that might happen. So with that, I think 3D printing is really opening some of the frontiers for physicians to be able not just to communicate between each other, but also communicate with a patient. And then I'm gonna pretend this is because Jeff mentioned it, but I did want to highlight some of the other efforts that the 3D printing community has done, especially over the last year with the COVID outbreak. And it's been very interesting, it's been very exciting, it's been very humbling, I think, from a lot of the perspective, but also this is the highlight that I really wanna make, that it does make a difference. This is an example from the NIH website, and this is the website that was specifically created with a collaboration between FDA, NIH, and the VA hospital to be able to look at the 3D prints that people are putting out and making sure they're validated and making sure they're safe at the end of the day. And then what's interesting, and I think a lot of the team members on this call also know this, if we look at the time frame right around March over here, this is about the time that some of those initial designs got posted by UW team. And frankly, the first two designs that were clinically approved designs came out of UW or UW and VA collaboration. They were a face mask, or sorry, face shield and a face mask that were collaborative efforts. And you can see this huge spike in the number of people who visited. It actually crashed the site at one point in time because it couldn't keep up. But it does make a difference in what we're doing. So at the end of the day, I think the last two talks highlighted a lot of the things that are being done and that people are thinking about. And I think we'll see more. And I really like to think of this as we're just stepping out. We're just stepping out of that fear zone into the learning zone, and we'll still have a bunch more to learn and a bunch of things to think about. Some of these examples people have already started doing. This is an example of taking the CT data sets and maybe look at them a little bit closer, trying to look at not just that segmentation aspect of things that I've talked about in the beginning, but maybe dive down a little bit closer and look at each individual voxel, that Hounsfield unit, and say, what does that mean? How does that correlate to the anatomy in question, and how can we print it so it's biomimically correct and we can actually use it? Here's another example of the work that was done within the University of Washington in taking a look at maybe more of the minute aspect of things. So this is an example of a chronic total occlusion, which is a blockage within one of the vessels in the heart, so a fairly small size, and thinking about how can we print it, and frankly, how can we bioprint it? And another one, how can we create training tools for physicians to be able to do that, which were done with a collaboration in the chemistry department and looking at some of the hydrogels that were widely discussed during the previous couple talks. And that will be it for me.
Awesome. Thank you, Dimitri. Let's give Dimitri a round of applause. If anyone has any questions, feel free to put them into the chat or into the Slack. But this is very, very cool. What do you see as kind of the main challenge in terms of getting surgeons trained with these 3D printed models? Is it the consistency of the material fill relative to anatomy? Is it the fact that the anatomy hopefully isn't bright primary colored like so many models? What are kind of the current gaps in need?
I think the biggest gaps and the biggest feedback that we've received from clinicians is we really have challenges with the materials that are traditionally used for 3D printing. A lot of the things are frankly hard plastics or hard materials. The elastomeric materials are still something that is going out on the market. And a lot of the times, as described, we're really dealing with the micro scale, and physicians for treatment are not necessarily interested in that scale of a sub-millimeter. They might be interested in millimeter or more in order to understand what's going on. And that transition of materials, and frankly, multi-materials that are durable and could be used in such a fashion that you can cut it, you can deploy things in it, you can sort of understand the forces that are involved, has been the biggest challenge that I think a lot of both the research community has been working on, but also the industry has been working on. So it's really interesting to see part of those collaborations that are transitioning into the clinical environment for people to actually use those parts.
Awesome. All right, so we're running about 10 to 15 minutes behind. And so I really want everyone to get to see Kelly's talk. So thank you, Dimitri. Let's give you a round of applause. And next we'll have Kelly Stevens. Her lab is developing new technologies to assemble synthetic human tissues. We've seen a little preview of what she's been working on in Cole's talk. And her research seeks to hijack and rewire aspects of nature's developmental programs to control the processes by which cells assemble to form human structures. To do this, she uses diverse tools taken from stem cell biology, tissue engineering, synthetic biology, micro-nano fabrication, and 3D printing. She is a UW alum and spent some time at MIT doing her postdoc, and has had some really excellent results recently. So thank you, Kelly, for giving us this talk today.
Thanks for having me. It's good to see you all. Thanks for being here. My kids are going to be getting home in like 2 minutes. And so if screaming happens, I apologize, and I might whiz through some of this. Just so you just be forewarned. By the way, nature is the most amazing fabricator. If or when y'all have kids someday, just remember how amazing that is. My lab, I'm an assistant professor at UW. We're working on 3D printing human tissues, among one of the many things that we're doing. The organ that we're particularly interested in is liver. It's the biggest visceral organ in your body. It's really awesome because if you cut out a piece of it, it can regenerate. Problem is that if you injure the liver through a bazillion different kinds of injury methods, everything from alcoholism, which is the one everybody always thinks about, right? But to other genetic diseases like even cystic fibrosis and literally hundreds of genetic diseases, many, many different viruses, hepatitis C, hepatitis B, and on and on. Anyway, if you injure the liver, it loses its ability to function and regenerate and everything else. So our pie in the sky goal is, can we 3D print a liver? Our thinking is if we can 3D print musical instruments, clothing, spacecraft, cars, everything else, why can't we 3D print a liver someday? Pie in the sky goal, right? I would be remiss to not mention my close friend and collaborator, Jordan Miller, who is my partner in crime with all of our 3D printing work in the lab. We just have a super tight sort of, it's almost like a shared group, to be honest, on all of this work. So I just want to make sure that I highlight his major, major, major contributions here. He's really the guy that developed the initial biomaterial formulation that allowed us to do some of the stuff that Cole actually mentioned in his talk, so I'll kind of whiz through it. It's all based on stereolithography. So for those of you that are not familiar with the basic idea here is you have a bath of liquid or something, it's sort of a viscous liquid, bath of liquid, you flash light on it, and wherever you flash the light, it turns into a gel. Okay, so imagine it going from liquid to jello. But what's cool about it is as you do this, if you flash with a projector, you can get different patterns in your different layers, right? And so you go flash, flash, flash. As you move your thing up and down, your part up and down on in the Z direction in a stage, and you get these different complex structures emerging, right? So that's been in the 3D printing and fabrication spaces for a while. You know, the part that was tricky was to get materials that are compatible with human cells and also are compatible with making parts that are really deep or thick, okay? I won't go into the material nuances of that. That's really Jordan's area. I mean, I can talk about it briefly if you all want to hear, but here's the basic idea. Maybe you can get a 3D tissue or organ, engineered organ, to emerge out of this bath. Okay, so these are supposed to be the blood vessels in this lung structure. So as Cole already showed you, and I won't go into this too much, we've been able to 3D print structures that are more complicated than had been possible previously. That said, as he also mentioned, the resolution is super limited, and so we're not there yet. We have been able to improve the resolution so here in this alveoli model, which is like a part of the lung, right? These sort of struts that you see, or the vessels that you're seeing here, about 300 microns across in resolution. We've been able to get that down to about 50 microns now. And so just with more materials and actually both hardware and software innovations, kind of like what Cole was talking about. So this is also coming, improving. So our, this way of doing things is much, much faster than Cole's way. So their limitation is speed. Our limitation is resolution, right? And so hopefully these will start to find some happy middle. So I said that one of the magics of the secret sauce that we're using, which is really similar to the materials actually that both Cole and Albert already introduced, and so I won't go into the chemistries. These are all based on polyethylene glycol diacrylate, as well as some mixed in with some proteins in the body called gelatin, collagen, that's all mushed up. And because of the sort of formulation, we can actually just, as we're printing in the bath, we can put cells right in the bath of the liquid and print right, essentially freeze them into place as we're printing, okay? So here we've got a printed structure that has both endothelial cells in it. These are the cells that line your blood vessels. It's also got hepatocytes, which these are the cells that perform almost all of the 500 functions, all of the sort of chemical processing functions that your liver does. We've been able to show that we can implant these printed tissues into mice, and they survive, and they have some liver functions. We can get them to actually grow their own blood vessel networks now. This is some of our newer data. And we can get, again, these human liver cells to survive in these tissues. And so it's sort of a step. We're still far from 3D printing a human liver, but we're stepping in that direction, right? One of the things that I think has been kind of fun about this, and also I think that we as people that are trying to print human organs need to start to get real about, is how nuanced the body really is. So I'm just going to give you one example here, and we can think about it together. This is moving a little bit out of the fabrication space, a little bit deeper into biology, which is, by the way, where my lab tends to sit. We're sort of more on the biology side of these things. So if you were to, so this is what the liver looks like from the boring zoomed out view, right? But if you zoom in and look at it, so this is about 1 millimeter across from here to here, okay? Or even 0.5 millimeter, depending on what species you're looking at, mouse, human, whatever. And these little peach cells here, whatever color you call that, are called hepatocytes. Those are the cells that perform all the functions. And in the schematic, they look all the same, right? So all of those hepatocytes, the one over here looks the same as the one over here, right? They're all peach. But in reality, they're not the same in the human liver. And in fact, that nuance is super important. So in reality, if you took this hexagon and you looked at it, and these were all now hepatocytes, say these are the peach cells, I've just now made them on a gradient from red to white. Actually, these cells, even though they're the same type of cell, they're actually super, super different in the jobs that they do, okay? So I said the liver does 500 functions. Well, not every cell does every 500 functions. Instead, they divide and conquer, okay? And so they're actually spatially orchestrated or at the sort of genetic or molecular level. So even though they're the same population of cell, they're doing different stuff. Okay, and this is like super, super spatially organized, which I think is interesting for fabrication people, right, in the liver. And so what we've been trying to say is, how can we not only just organize where cells are in the liver and 3D print their location, but also 3D print what they're doing, okay, what jobs are they performing, and make that also mimic human organs, right? And so the way that so our thought is, can we essentially mimic this radial distribution, molecular distribution in the liver? So how do we do that, right? So what we realized, actually we were really inspired by Cole's work in this space of optical patterning. What we realized, if we can 3D print fluidic networks into these tissues, there's a lot of ways to pattern stimuli, such as, for example, heat or thermal energy into tissues using heat exchangers, right? So if you're an engineer, you're going to do all kinds of work in heat exchangers, meaning you put hot fluid through a pipe and you can change the thermal distribution around that fluid, right? We said, okay, well, we know that our hepatocytes have certain genes that turn on and off in certain spatial locations. What if we genetically engineer our cells so that they activate genes in response to heat? Then in theory, we could get these cells to activate the genes that we want them to activate around just the channels, okay? So it's this combination of, we're really bridging this gap between actually fabricating stuff, now fabricating stuff to make cues drive the cells to do stuff that we want, okay? The functions. And so here's what our setup looks like. I'm going to kind of whiz through these next ones. We've got our bioprinted tissue, and now instead of just injecting dye into it or cells into it or whatever for making our blood vessels, instead, now we're injecting hot fluid into it, okay, to make these what we're calling thermofluidic networks in the tissue. So they're basically heat exchangers within these bioprinted tissues, okay? So what you can see here is the fluidic pipes that we printed. And then when you just look at this with a thermal camera that measures temperature across the surface of the tissue, you can see that we get a gradient of temperature distribution across these tissues. We engineered the cells, I'm not going to go in this, to respond to heat, okay? In other words, if you turn up the temperature, all of a sudden the cells will activate the genes that you want them to activate, okay? So genetic engineering again. And then we showed that if we then put these together, so now we put our cells in our printed tissue, we put fluidic networks that are hot around them. Basically what we were able to show was that if you just make different heat exchanger profiles, you have a co-current where they're going in the same direction, countercurrent where they're in opposite directions, blah, blah, blah. You can change the heat distribution across the gels, and you can also change their genetic, the genes that they're activating across the gels, okay? So we're doing both structural patterning with the fluidics, and we're also doing molecular genetic patterning by stimulating the heat system. We can do this dynamically. So my grad student thought he was being cute, and he made a clock, and he stimulated on day 1, 2, 3, 4, and it essentially walks around the clock, right? Which is exciting for a biologist. And finally, I'm not going to go too deep in the biology, but just show you that we're not just trying to play and make pretty pictures here. It's well known that there's a molecular signal in the body called Wnt, and it's kind of the master regulator, it's the boss, okay? It says if Wnt is in a gradient, I'm going to tell all of those other 500 genes or molecular cues to sort of walk in line, okay? And so in other words, if the boss, is properly organized, all of the other ones will automatically properly organize themselves. That's sort of the working hypothesis amongst biologists in the field, all right? So he said, well, if instead of activating this rainbow that I was showing you before with heat, what if instead we activate Wnt, which is this molecule, right? And so I'm not going to go into the details again with the genetic engineering, but we made these heat constructs, these genetic constructs where we can activate all of these different biology factors in the body, the Wnt pathway, and we show that we can pattern them. And then we're now doing studies that are starting to show that we can get the rest of the genetic signals and molecules in the liver to sort of walk in line in response to that. So we're starting to get these zonated profiles, or spatially localized gene expression profiles in our engineered liver tissues. And I'm going to stop there and say, by the way, happy International Women in Science Day. I'm not sure if I got that right, or Girls in Science or something, Women in Science. This is what Twitter is telling me. Thanks again for Jordan Miller. He's just an amazing collaborator. I want to make sure I acknowledge him when I give a bioprinting talk in particular, and the funding. And thanks to y'all for inviting me into this cool and exciting community. I'd love to take any questions if you have any.
Awesome. Let's give Kelly a round of applause.
I think I saw a question from Subhasha.
Yeah, I had a question to Kelly. I was wondering like in the past decade has seen a lot of growth in 3D printed ovaries, artificially creating womb-like environments to build non-mammal to reproduce non-mammals. I was wondering if could we really think about 3D printing uteruses in the near future? I mean, it's purely from an academic perspective, thinking about how many lives are lost on a regular basis because of pregnancies in third world countries.
Yeah, I can tell you that is an area that people are thinking about. How far it is, I think that's, of course, gestating a human is very complicated, right?
Yeah.
And so I would say, am I optimistic that I would guess there's going to be a lot lower hanging fruit, right?
Mm-hmm.
Is it
yeah,
possible? Is it possible? I would guess that it is possible. I just would never venture a guess as to when. And I can say that people are thinking about it. Other bladders, for example, is also sort of a circular type thing that's easier and maybe a little bit less, if you mess it up, it's not quite so bad, right? And I guess we could argue that our bladder is super handy to have, so there are some things that I think 3D printing will be more apt to solve sooner than later, sooner than uteruses, but it's definitely an area that people are thinking about. And actually just female reproductive biology broadly, yes, is an active area of research. Although I would also argue I'm a part of a huge faculty group in our profession that's mostly composed of women. And there is always so much discussion on that group about how much women's health issues are really understudied and how we need more women engineers so that changes. So, this is an interesting area for sure.
Thank you.
Sure.
Okay, do we have another question? If not, I will use my prerogative to ask one. So the bladder, I think, is a great example, right? We originally started off 3D printing bladders, and then, correct me if I'm wrong, Anthony and Tola just started casting them and coating them in balloons.
Right on.
And so is it, one of these cases where like, in the rest of engineering, 3D printing is always the second best solution? Is that really the same case in biology, or does the need for complexity really make it so that 3D printing is going to remain the best choice for structuring these patterns?
I don't think that it is the best choice for most things right now. Does it have the potential to become the best choice in the future? Absolutely. So the big challenges are size and scale, in my opinion, and material. And so all of these things, like if we can overcome, for example, Cole was mentioning speed, that is absolutely essential for biology, because if you don't pattern with speed, all your cells die. Okay, and so he sort of minimized why it matters, but the problem is that until they get that, it won't be useful from like a building organ standpoint. And the same with our technology. There's just these little details that need to be solved still. Things like what material, how to get the cells to interface like you want it to, and how to do the patterning and blah, blah, blah. But I really do think that I used to be pretty skeptical about 3D printing, to be honest, and whether it would actually be useful for biology. And I have to say I've become a believer. I just would never say when. Okay, like I don't want to say it's going to happen in 3 years or 10 years or 50 years, right? But I do really think it's possible. And I think that 3D printing is by far one of the most interesting and useful technologies for especially fabricating really complex organs, like, for example, the liver that I was looking at. Simple stuff, sheets, things like skin, circles like bladders, probably you can do it a different way. But the really complicated things, I don't know how you're going to do that with other technologies. And I should say my lab does a lot of other technologies in addition to 3D printing.
So what do you think was your conversion moment towards 3D printing as being a viable tool instead of just smoke and mirrors?
Interestingly, so I showed you the stereolithography stuff today, right? Cole showed you another piece of work that I've done with Jordan Miller, the sugar stuff. That was when I had the moment. I think that technology actually, in many ways, is more exciting and more interesting, because I said that one of the big Achilles heels is the materials. And I minimize this, but the materials that we use for the stereolithography are not particularly biologic, like the cells don't love them. It's okay, it works okay, all right? But the biologist in me is like, not so sure, right? But that particular sugar technology, you can use any material with it. And that leap alone, meaning any, like, you know, materials from the human body or whatever, right? And so I shouldn't say any material, but many, many more. You just completely open up your portfolio of options. I actually think that that paper should have been cited a bazillion more times than the science paper. So, you know, whatever, people can, you know, get excited about what they want, but I can tell you that that was the first time that I was like, oh, maybe we could do this. So, yep, we're still moving forward with both, right? But it was the first time I became a believer. So.
Okay, any other questions from the audience?
And that's laser sintering, by the way. So completely different technology, right?
So then I guess the question is, what can, you've got such broad collaborators. Actually, I'll open this up to the entire panel of speakers. You have such broad collaborators that you've already been working with. What are some of the missing links that you think you would want to see imported from other fields that other researchers could be developing to help you with your research? I think we got a strong sense of that from Dimitri with the need for multi-materials and better digital processing. But, you know, if you could just throw it out, you got your MacArthur Genius Grant, which I'm sure is coming, and said, I'm going to throw it down to have other researchers build me X. What is X?
For me, it would be, there's two things, and my lab is now working on one of them. One is, we don't know what human organs look like. We pretend we do, because we sort of know what mice look like, and then we have some level of information about human organs, but actually we don't have maps of human organs. So the question of what do you actually 3D print, we don't actually know. If you wanted to actually mimic the structure and also molecular detail of a human organ. And so I should say that's a hugely active area in my group right now, is just building better blueprints. So that side for sure. So I'm partnering with people in mechanical engineering, genome sciences, blah, 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 to build better blueprints. And then for my interests, we're already doing that. So I wouldn't say that's what I would personally ask for, but the part that I would personally want that we're not doing as much is more of the gene engineering that I mentioned, the synthetic biology. I think to actually build human organs, we're probably going to need to use nature's help a little bit. Nature is very capable of building entire human beings, in fact. And so if we can get a little bit better at deploying these technologies in tandem, like better genetic, I showed you just like the simplest tip of the iceberg. You turn one gene on in response to heat, right? But if you could have like better, interesting portfolios there, that would be huge, in my opinion. So I think it's not necessarily just from the 3D printing side, but like you said, a blend of the fields that will get this done. I'd love to hear my colleagues' thoughts though too. That's just my 2 cents.
Cole and Albert actually had to step out. I don't know if Dimitri has anything to leave, but Kelly, you'll have to find those answers offline and then share them with us from Albert and Cole.
Yeah, I guess from my perspective, I'll just re-emphasize one more time and maybe just expand a little bit more. I think the idea of a better blueprint even translates to my side of things. And for me, it's the idea of unlocking that data sets. I know we'll look at them on a daily basis, but do we truly know what they mean? I know this is part A and what this is part B, but I think we could do a bit more digging in what each one of those parts actually represents in the physical world, not just the digital world. So it would be good to see a little bit of that, I guess, translation and language to go from the DICOM to the physical aspect of things. And then better materials, I think, is not just about the multi-materials. It's about the work that everyone in this group has been doing in regards to how do those materials change over time. when you introduce stress to them, when you introduce heat to them, when you introduce blood to them and other aspects that are actually going to change how particular devices being deployed or anatomy being challenged are going to change in their response.
Awesome. All right, well, I feel like the audience has some homework to do and to go build better materials and devices. So let's thank our panelists who are remaining again and all the ones who had to go early. And I hope you guys have a wonderful evening and enjoy the snow.
Take care, y'all.